Today we are going to talk about the final part of this course, which is analyzing RNA sequencing data, actually doing stuff by ourselves, how we can look at biological data and analyze it, and RNA sequencing data would be the best, according to me, to learn, hands-on practically how to do stuff. So the first thing you should do is that I have sent you all a mail carrying a particular file. You just go ahead and download that file quickly. Please, using a thumbs up or something, tell me if you have done it. We would be using this file for all the analysis today.
So where have you sent it? Mail.
Mail, yes.
So sampling and estimation কত?
Na, I have just sent you a mail. Raw data all counts.
Sir, in CSV format, right?
Yes, in CSV format.
Yeah, it's done.
Okay. I will also send it in the WhatsApp group in case you guys are not able to get it.
Yes sir, got it.
Everyone has downloaded the file. Anyone who has not been able to? Okay. Next, what you do is that go to our studio and start a new notebook and write these lines in. Install packages BioC Manager, and BioC Manager install DESeq2, and then call the libraries appropriate. This will take some time to download, so I want this to start off like you start off with it, and then we will discuss about RNA sequencing itself.
স্যার চ্যাটে কপি পেস্ট করে দেবেন একটু?
Okay, I hope everyone was able to do this, run this, and it should install both the BioC Manager package as well as the DESeq2 package. So while that is happening,
Sir, sir, can you wait for a bit? Like mine is not done till now.
yeah. I am telling you, it will take some time. So let it go.
Okay, okay,
We are
okay,
going to discuss something else, and we will come back to it.
okay, sir.
Okay. I hope this PPT is visible. We are going to quickly learn a bit about molecular biology. So yes, I think everyone here has an idea about what the central dogma of molecular biology is. It is that DNA makes mRNA, which then makes protein. The process of going from DNA to mRNA is called transcription, and the process of mRNA to protein is called translation. And the idea is most of the gene regulation is done at this level, the transcriptional level, because transcription by itself requires a lot of energy. Each of the additions of, like, just forming the mRNA requires a lot of ATP, and this is the first step that the cell will try to block any kind of non-required aberrant transcription, because if you don't want to make a protein, why would you even make an mRNA out of it? So most of the gene regulation is at this level, DNA to mRNA. So transcriptional level gene regulation. So DNA, RNA, protein. Transcription is DNA is transcribed into RNA, translation is RNA is translated into proteins. Major gene regulation occurs at the transcriptional level. I think everyone would be okay with this. We will be talking a bit about what these guys in the middle are and how important these guys are, the mRNAs. So I will just start off with an idea that during development we know that different tissues develop from initially a similar number of similar looking cells. So in the entire body develops from the single cell called the zygote. And the important thing to know here is that the DNA or the genomic material of all cells throughout your body, doesn't matter they are present in your hand, your heart, the retina of your eyes, all the genomic material, the DNA is absolutely the same. So now the question occurs, how is, if the DNA is the same, how does your eye develop differently from your heart? So why these cells function differently, form different types of tissues, would have absolutely different characteristics and functions? How does that occur? So if they have the same starting material, how do they end up being something so very different from each other? So you have blood cells, you have macrophages, you have bone cells, you have so many different things, but same starting material. How would they, all of them with the same starting point, diverge so far away from each other? The answer is called differential gene expression. The idea is the entire genome or the entire DNA is not transcribed. You have like 30,000 genes present in the DNA, but you're not transcribing all the 30,000 genes all the time. These all cells have something called gene expression, which is like, you from the DNA you make mRNA, you make protein. But different cells can express different mRNAs from the same genome. So your heart is expressing a separate set of mRNAs and thus a separate set of proteins, and doing a very different function from the cells present in your eye, which are also expressing a different set of mRNA from the same starting material. It is just picking and choosing what it wants to express, and then the mRNA is translated and the proteins then do all the function, and you form the eye, which is very different from the heart and which functions very differently. So this is the idea behind differential gene expression. Same genomic DNAs, but this genomic DNA is transcribed differentially to transcribed differentially, so it gives rise to different sets of mRNAs which are unique to each cell type and which causes them to be different from each other in function and form. This can also be looked at in the disease context. So, in case of cancers, you have differential gene expression as well. In the normal case, say you had a gene A which had 3 copies of mRNA being transcribed, but in case of tumor, this or cancer, this particular gene A is somehow dysregulated and forms multiple, much more copies of that particular mRNA, which then forms a lot of the protein and then which gives rise to tumor or cancer. Or the other way around, in which case in case of gene B, you had 3 copies of transcripts or mRNAs, but in case of the cancer situation, these mRNAs are either not transcribed or they are transcribed but are dysfunctional, that is, they cannot form the proteins. So here also you have an example of differential gene expression. Even with the same starting material, somehow the mRNA levels are different from each other, and the lesser amount of mRNA here actually causes tumor or cancer growth. So on the left side, these kind of cells, these kinds of mRNAs, which when they are overexpressed or upregulated, that cause cancer, are known pro-oncogenic or oncogenic genes. These are ones that are regulators of cancer and prevent cancer growth. These include genes such as P51, which stop cell proliferation when it is getting out of hand, but when you have dysfunctions in them, then cells can grow however they want, and that causes cancer to actually arise out of a tissue. So essentially what I am trying to tell you is that differential gene expression is the key to variability in all cell types. So cells are different from each other because their mRNA profile is different from each other. So this is known as the transcriptional signature. So essentially all the mRNAs, the sets of mRNAs and how many mRNAs they are transcribing at any moment is known as the transcriptional signature of a cell. So this transcriptional signature tells us a lot about the state of the cell, if the cell is functioning normally, like in these cases, or say the cell is going to proliferate. It says I am going to start dividing a lot. That also you can find out from the transcriptional signature. If it is going to differentiate into a different cell type, the same cell says that one part of me will go and form erythrocytes or RBCs, the other part of me is going to go and form white blood cells. All of these can also be figured out from the transcriptional signature. And finally, you can actually try and figure out if the cell is in a disease state or not. Is it proliferating too much? Is it making too many nutrients? Is it consuming too many nutrients? All of this can easily be found out by transcriptional signature, and that is why looking at the mRNA state of all these cells is very important and is an extremely powerful tool that essentially everyone is doing nowadays. So any kind of research you open up, you will have RNA sequencing somehow or the other getting into it, because everyone is very interested on this idea. So transcriptional signature gives us a lot of information about the cell, and it can differential gene expression analysis can tell us how the cell is different from a normal cell and what exactly it is trying to do. So just a quick example of it. So the process of formation of different types of blood cells is known as hematopoiesis. But all of these cells which are down here, the neutrophils, the eosinophils, the basophils, platelets, RBCs, the B cell, the T cell, the T cytotoxic cell, everything that you see in the blood, all of it comes from one parent cell. There is this cell known as the hematopoietic stem cell, which will give rise to all these. So these do not appear out of nowhere. There is a sequential differentiation of this hematopoietic stem cell, which first forms one type of progenitor, and like two different types of progenitor. One will be the red blood cells progenitor and the other will be the white blood cells progenitor, which then further differentiates into different types of cells. At each of these states, the all these cells are expressing different sets of mRNAs. To stay in the hematopoietic stem cell as a hematopoietic stem cell and not go further down the lane of forming blood cells, this HSC will preserve its mRNA transcriptome and keep on just expressing those genes, and it is very happy being a HSC. But the moment it has to go on and form these next set of progenitor cells, the transcriptome profile will change, it will start expressing different mRNAs, and the cell by itself will start to change and proliferate and form these two guys. These guys will then proliferate even more, forming the first set of progenitors. So you can see granulate monocyte progenitor, eosinophil progenitor, erythrocyte progenitor, and all of these cells can then by themselves change their mRNA transcriptome again to give rise to matured blood cells. So essentially, looking at the transcriptomic landscape of a cell, you can actually say what the cell is going to do, what the state of the cell is, what markers it is presenting, is it actually a red blood cell or has it not matured yet. All these questions can be answered using just looking at the mRNAs that a cell is expressing. So that is why this is such a powerful technique to use in research, that you know everything about the cell just looking at the mRNA. Everyone fine with this? Okay, so
Yes.
yeah. So, but how exactly do you look at the mRNA? You do something called RNA sequencing. So I think I think most of you know and have an idea of what DNA sequencing is. You can read the number of the base pairs that come in the DNA and know the DNA sequence. Something similar is known as RNA sequencing. A much more complicated approach, but you can actually look at all the RNAs that a cell is expressing. So using RNA sequencing or transcriptomics as it is known as, it can capture the quantity and the sequences of RNA in a sample. We are going to deal with this, which is known as bulk RNA sequencing. So you look at any tissue or any set of cells, you take the entire thing, crush them up and extract the total RNA out of it and then sequence the RNA. So it gives you average expression of what kind of genes were expressed in the entire cell population. So applications are obvious. You can do something known as gene expression profiling, know what genes the cells were expressing, and you can also do the differential gene expression analysis, in which you find out, yeah, so my cells who were in condition 1 were expressing this set of genes, whereas my cells in condition 2 were expressing a separate set of genes. So the difference between them will actually tell me more about what is different between the condition 1 and the condition 2. So you do these kind of analyses on the RNA sequencing data. So today we are going to look at the easiest form of sequencing and the easiest form of data analysis for RNA sequencing and transcriptomics, which is known as bulk RNA sequencing. There are multiple other ones, and I will tell you about it like later. Okay, so how do you do RNA sequencing? Here it is a very simplified diagram because I don't want to get much into it, but I will just give you an idea of it. So as I said, say you were looking at cells, you were treating it with drug A and drug B, and you were trying to figure out what the difference between the cells is. You can just do an transcriptomics and find out what the difference is. But how do you do it? You first take all the cells and then crush them up and take all the RNA out of them, and you isolate these RNAs from the different cells. So you have one append off, which is full of RNA from the entire cells that you have been looking at. You fragment the RNA, break it up, and then do a reverse transcription reaction. So you probably have heard of it, that there is an enzyme known as reverse transcriptase which can form DNA out of RNA. So even though the name says RNA sequencing, we actually never sequence RNA. We form a cDNA out of it using reverse transcription, which is the first strand synthesis, and using the first strand we do a second strand synthesis and then do a PCR to amplify the all of them. So you essentially you have the sequence of the RNA just amplified and in DNA form, and then you do something called ligating different adapters. You add different sets of DNA fragments to each of the ends, which helps in doing something called the next gen sequencing. You can use the Illumina platform or the Nucleopore platform, and you know what the sequence of the DNA is, and then from the sequence of this DNA you go back and find out the sequence of the RNA that you had. Just this last step is a bit more complicated than it looks because you have fragmented the RNA. So essentially imagine you're taking an entire word and you're picking and choosing some letters out of it and finding what the letters are, but you have to put them back into context to make the entire word out of these fragments. So there is a lot of computational parts associated with this final part of the analysis, but essentially after all of this you will get which RNAs were expressed and how many of the RNAs were expressed in the sample.
Sir,
Yes.
can you just explain the last three steps once again?
Last three steps. So first, till here you have formed the DNA out of the RNA fragment. So you have done one reverse transcription step which forms the first set of cDNAs, and from the cDNAs you do a PCR to expand the amount of cDNAs you have so that you can have you can sequence easily. It is not a problem. Next, what you do is that there are DNA fragments which are called adapters. So these adapters have sequences that I know, and so these are just added into the mix, and then you add something called a ligase which adds these adapters to the ends of the newly formed cDNAs. To here and here you can see the adapters being in pink and orange, and this blue was our cDNA that we made in the third step. Now the interesting thing is that because I know the sequence of the adapters, I can perform the sequencing very easily, because for sequencing you need the DNA molecules to attach or base pair to another set of molecules, and then you do the sequencing. So that you do, so using the adapters helps out a lot, and then you just perform next gen sequencing. So I imagine that you people have gone through Sanger sequencing, how that was done. So this is just a much more powerful approach that actually does it. So you just can sequence a lot of DNA at the same time, and you can have multiple repeats of it so that you have a good idea. And from this also you get the same chromatogram that you get from Sanger sequencing, essentially the sequence of the DNAs that you were trying to find out. And after you have all of this, essentially this is just, so the entire mRNAs that you started with, these guys are probably around 1 kilobase or higher, so 1000 base pair or higher, longer. But after you fragmented them and did all this sequencing, you only probably sequenced 50 base pairs. So how do you put, where does the 50 go? So it is like you have 1000 letter long sentence, and you have been picking and choosing 10 or 15 letters at a time and finding what the letters are, but you have to still figure out where those letters go in order to make the total sentence, right? So that part comes next, where you put all of these sequencing information, whatever information you got, into a library. This library contains the entire words. It knows what the entire word should look like, and you keep on giving it smaller and smaller fragments of it, and it puts them all together into one, essentially reforming the sentence, and that sentence is the RNA that you initially started with. So essentially, like a jigsaw puzzle, it will piece together all these smaller RNA fragments and form the entire RNA, and then I know, yes, this is the RNA for the gene A, and I say, yes, I see there were 10 transcripts of it, so that information gets stored. So this is how you do the final assembly of the entire RNA sequencing data. Is that okay?
Yes, sir.
Okay, so I am not going into next gen sequencing, so it is pretty cool. You can easily find multiple videos on the Illumina platform on how they do it. If you guys still have any problems, then I can tell you more about it, but it is just sequencing. The DNA, what the DNA sequence it is. That is all we care for now, because we are looking at it from the statistics point of view. I just want you to get idea that differential gene expression analysis is very important, and you do it via transcriptomics, where you get a data set out of it. That's all. So the output file will be a .csv file, which is a comma separated values file, which looks very complicated, or a Excel file that also looks complicated. It just has names of different mRNAs and how many transcripts or copies of the mRNA were being expressed in different conditions. What you do is with it is up to you, which is we will learn now how to do it. We will look at all the genes, and we will try to find the differentially expressed genes. So the idea is, a normal cell was always expressing some amount of mRNAs. A disease cell or a cell under a different condition is expressing a different set of mRNAs. But originally speaking, most of the mRNAs will be the same. So it was say cell A was expressing 100 mRNAs. Cell B will express 90 of the same mRNAs. It will just start expressing 10 different ones, or different levels of 10 different mRNAs. Those 10 are key to why that cell B is either diseased or is under a different condition. So that those 10 are the ones that we want to find out, and you can't go through and look at all the data, because there are 25,000 mRNAs being expressed, and we use different tools to do it, which we will be doing today. We will using the DE-seq2 tool to actually find out the differentially expressed mRNAs and differentially expressed genes thus, and find out which genes are doing what in the entire cells. Is that okay with everyone? Anyone not following, please
Yes,
tell
sir.
me. Okay, so we will actually go back and look at our RStudio. I hope this has run properly for everyone. It should just go green without any errors if it is running properly.
It's still buffering, sir.
It is still buffering.
Yes, sir.
Eh?
Yes, sir. Same.
Akbar, someone show me their screen and tell me what is happening. Why is it still buffering? What is happening?
Sir, can I do?
Do.
Acha, wait. There is one part. One part it asks me if you want to keep on updating all the data, or yes or no, or all. It asks one question. Maybe it is stuck in that. Check in the terminal.
You have to press A or N or Y. Press A. It will not do anything without you pressing anything.
Sir, terminal দেখাচ্ছিল।
টার্মিনালে এখানটাতে আসবে, there will be something terminal কনসোলের মধ্যে এখানে আসবে, it will ask you the question, do you want to
So I pressed A. A করতে হতো নাকি
everyone
Y
just
by
press A. I also had this somewhere, it is now lost, but yeah, you have to press A, otherwise যে এমনি করে আসবে update all or some or none packages, press A and it will update, and then it will quickly work through, like go.
Sir, it's done.
Okay, everyone, anyone who is still stuck, please tell me.
স্যার হচ্ছে
এই করে নাও, তারপরে we let us start.
ইয়েস স্যার ডান
ওকে, সবাই হয়ে গেছে? এনিওয়ান lagging behind? Okay, I hope not. ঠিক আছে, next what we do is that I hope everyone has downloaded the dataset that I sent you. It should be raw data all counts.csv. So you just in order to load this dataset, what you have to do is that write this line down. We will name this dataset mutant data, mut data, and read csv is the function that is used to read it, and then file followed by the name of the data. But since you probably have downloaded it somewhere weird in the downloads folder or something, what you will do is that তোমরা ফাইল খুলবে, go to the file, go to downloads, say this is what I wanted to download, and press here, or copy as path, and copy as path করার পরে you put this in this location, so it should look like this, and once you run that, it will open the file. Just go downloads folder.
Excellent.
Go downloads folder, look at the date, find the file, right click, copy the path, put the path in the file wala double quotes.
স্যার একটা কোয়েশ্চেন আছে স্যার, আর এ কিছু কমান্ড আছে না যেগুলো টু ভার্সন আছে, যেমন জিজি প্লট আছে, জিজি প্লট 2 আছে, সিমিলারলি রিড সিএসভি করতে গেলে রিড সিএসভি 2 ও দেখাচ্ছে, তো ওই 2টার কি ফাংশন? ওটা কি জাস্ট এক্সপ্যান্ডেড ক্যাপাবিলিটিস?
It is just a updated file which can use more csvs, but for us this is we can use just the older one. So most of them, ggplot and ggplot2, ggplot2 is just the updated version of the previous one. deseq2 is the updated version of deseq. We just use the one that works.
Sir, mine is showing an error. Can you see
Yeah.
one
Show
time.
me your screen. What is the error? It could be that the backspaces are the wrong way around. Just show it to me.
স্যার 6টা রো দেখাচ্ছে।
Sir, can you see this portion?
I can't. I can. Okay. You slash you. Okay. So what you do is that wherever you see that slash symbol,
Yes.
ওইটাকে just delete it. The slash symbol.
So the slash symbol is everywhere, right?
Yes.
So
You have to
slash
delete.
u is
So
okay.
what happens is that slash u gets read as something else. Don't remove all of them. You have to replace it with the reverse slash. It shifter badige hobe, which looks like a ulto diger division sign.
If,
Yes, like that. যত কটাতে error
oh,
দেখাচ্ছে সব কটাতে you have to change and then try.
okay.
R
Sir, one
does
second.
this sometimes, so it becomes a bit stupid. And run. Yeah.
Yeah, thank you.
Yes. হ্যাঁ বলো, অন্য কেউ কিছুটা বলছিলে?
স্যার আমি বলছিলাম যে মাত্র 6টা রো দেখাচ্ছে।
You have done head না, head only displays 6টা row.
ও
একদম
হ্যাঁ হ্যাঁ হ্যাঁ,
প্রথম
সরি
ক্লাসে
সরি, আমি
আমরা
ভাবলাম
করেছি এটা।
পুরোটাকে আমরা করি।
হ্যাঁ, প্রথম ক্লাসে একদম এটা দেখেছিলাম। when you want to look at a big data set, you don't just display the data set, it becomes odd to look at. You do head of the data set, it looks at just the first 6 rows, so you have an idea what is happening. Everyone else, you are also with me this far?
স্যার ফাইলটা আরেকবার দেখাবেন কি করে করতে হয়, তা আসছে না।
কি কোনটা ফাইলটা কি করব, কি
লাইক
করে
ফাইল
খুঁজতে
ম্যানেজার
হবে
থেকে
তাই না?
কি করে ওই
So
জিনিস।
you go to তো you have downloaded it, right? So you can go to your browser and যেখানে downloads folder, you will see the file here somewhere. যেমন আমার এখানে একটা অন্য ফাইল দেখাচ্ছে। Open the folder it is in. I got the file. Right click and copy the path. So find the file wherever you have downloaded, copy the path of the file. Once that is copied, go back to RStudio and যেখানে ফাইলের ডাবল কোটস, যেখানে আমার এখানে র অল কাউন্টস লেখা আছে, you just paste it, and after pasting, if it is giving you the same error as Dipanjali, just remove these slashes and replace them with the ulta ones, and then it should load.
ইয়েস স্যার, ডান।
Done. Anyone like falling behind, please tell me, because most of the analysis I want you to do it by hand yourself, so that তোমরা বুঝতে পারো কি করে করতে হবে. Anyone who is not able to open the file? Okay, so whenever you send your RNA for sequencing, this is the file that you should get. So any company, any research you are doing, you send for bulk RNA sequencing, they will send over a file such as this. So this file normally looks like this. Is a general idea of it. The first row most of the time is called the gene ID. These are all IDs of the genes. If you have been doing a bit of bioinformatics, you know these are ensemble IDs from Uniprot, a ensemble IDs from the EMBL dataset. So essentially this is the general ID for the gene, and then you have the gene name followed by how many transcripts were detected. So in the first mutant, so most of the time what you also do is that you send in replicates. So you just, so condition A, condition B, you just don't send condition A acta and condition B acta. You send it in replicates. You send condition A tinte sets and condition B tinte sets. 3 different experiments but under the same conditions, so that you can average out any problems that is occurring. So here in this data set we have, so when you are looking at any condition, say you have been giving a drug A and a drug B to the cells, then you don't send just drug A acta কতগুলো cells drug A তে একটা প্লেটে ছিল petri plate imagine, but you don't use, what we use tissue culture plates which look the same. So drug A ছিল আমার একটা প্লেটে, drug B ছিল একটা প্লেটে, তার মধ্যে cells ছিল. What you don't do is that you just take the RNA from these 2 plates only and send them. You actually do it in replicates. So for each of the plates you have another plate which looks exactly the same cells, you have put in the same treatment, but you do it 3 times, essentially just to make sure that if one plate doesn't have something odd in it, and that gives you a different result. So you repeat the experiments 3 times and you extract RNA from all 3 plates and send them. Here we have done the replicates 2 times, so same cells which are mutated for some reason. So in this particular data set we have a wild type cell and a mutant cell. That particular cell has something odd going on with it which we want to find out. So I have taken 2 different plates of the mutant cells, essentially 2 different replicates, and extracted RNA from it and sent essentially 2 replicates for both the mutant situation, the replicate 1, replicate 2, and 2 replicates for the wild type situation, replicate 1 and replicate 2. Essentially you are doing it twice so that kono error hole it gets averaged out. Fine?
Yes sir, বুঝে গেছি। আর জাস্ট একটা ছোট কোয়েশ্চেন, মানে এই ভ্যালু গুলো কিসের জন্য, যে আমরা মিউটেশন 1 এর রাখাই মিউটেশন টাইপ 2 এর জন্য, আর তাদের ওয়াইল্ড টাইপ গুলো আছে, ভ্যালু গুলো কি ইন্ডিকেট করছে? পজিশন অফ মিউটেশন
are essentially number of mRNAs. So PTPN12 gene, there are 506 mRNAs that it detects. So whenever transcription is going on, you don't just transcribe one transcript, you actually always have a lot of transcripts going around. So this just shows you have 506 of it in replicate 1 and 478 in replicate 2 in wild type.
Got it.
Okay, so this is just the number of transcripts of the particular mRNA. So this is what your general RNA sequencing file looks like. So now the entire idea is we have been doing something very similar. So we have been looking at say 506 versus 478. Is this difference significant? So these are differentially expressed genes or not? So we have done t-tests, we have done ANOVA, we know how those work. You just compare, you are essentially comparing these 2 quantities and figuring out if they are different or not, if the mRNA expression is different between, for this particular gene, between mutant replicate 1 and wild type. That is the entire idea. This, you can imagine it as a t-test. If you wanted to do it manually, you can do it for all the 25,000 that are there, do t-test for every one of them and then figure out if they are different or not. We just have a better way of doing it. The idea is just the same. You just compare the number of transcripts in condition A and number of transcripts in condition B and figure out the differentially expressed genes. ঠিক আছে? We will look at the same parameters, we will look at the p values, we will look at how different they are, we will look at the exactly same things that we have been looking at so far, just we are doing it using a tool called DESeq2. Okay, next what we do is that we have to prepare the data for DESeq2. As you remember, when we were doing some tests, when we are doing t-test, it only took the values as matrix. So we will have to do as matrix to convert the data set into a matrix instead of a data frame. DESeq2 also has a prescribed format in which you put in the data. So we will have to process the data. So this step is important and is involved in all the pipelines of analysis. So you have to make this is what you got, and you have to change it a bit so that it can go into the tool directly. So how do we do it? First thing is that the gene ID part of it, we don't want it as a column in the entire thing. What we want is that we want to this gene ID part of it to be as the names of the rows. So you remember we have used the function row names, in which we took a particular column and made them not part of the data itself, but the names of the data. So we use the same function here, row names, mute the mutated data, and mute data $sign gene ID, $sign just calls this entire column, and then we do head of mute data. Do it and see what do you get.
স্যার জিন আইডিটা দু জায়গায় দেখাচ্ছে, একটা রো এর আন্ডারে আর একটা আলাদা করে অনেকগুলো।
ইয়েস, ওটা দেখাবে। কেন কি আমরা রো নেমের মধ্যে তো জিন আইডিটা দিয়ে দিয়েছি, but we didn't delete this entire column. To delete the entire column, we will use the following command. So we are making a new dataset called mutedata.deseq, and that is a matrix as dot matrix, and that matrix is just mute data minus the first and the second columns, C minus C1 2. You remember that whenever we did subsetting in the first classes, minus of a particular vector would just remove those part of the vector or the data frame from the entire set. So this will remove the entirety of the first and the second column. So these two will disappear, and then you have the row names where they are, and if you do head of the entire DESeq dataset, it should look like this. So now the gene IDs are row names, though these are no longer part of the entire dataset, and the first and the second columns have disappeared. You just have the number of transcripts and the different conditions. করে দেখো কি আসছে।
স্যার আমার কোয়েশ্চেন আছে, এটা এক্সিকিউট করলে যেমন আমরা তো জিন আইডিটা দেখতে একবারই দেখতে পাচ্ছি।
জিন আইডিটা একবারে দেখতে পারছি মানে আরেকবার বলো।
না আমি বলছি মানে here we see the gene ID in one column only, right? মানে after applying the mute data dot desequence this command.
Yes.
But we are eliminating the first two columns, right? Minus
Yes,
I mean
but
minus
these are
one
now-
to minus two.
হ্যাঁ, তো এইটা না এখন this is, so you should see it like something like this first, if before you do the as matrix deletion. So essentially the gene ID column has been replicated as row names. So row names are no longer part of the dataset itself, but it appears as indicators. जो এখানে যেমন আমাদের 1 2 3 4 5 6 row names গুলো আছে, gene ID just is replacing that 1 2 3 4 5 6. See, the 1 2 3 4 5 6 has disappeared. Essentially ওইখানটাতে আমরা এই gene ID গুলো দিয়ে দিচ্ছি, and this is no longer part of the dataset anymore. It is just acting as this 1 2 3 4 5 6 এখানের মতো, and then we delete the first and the second column, which are these two. So now after that you will just be remaining with this and the numbers with the conditions.
Acha, got it. So here the gene name is just the row name actually. When it
Yes,
column
we are doing that now. We are setting it as the row names.
got it. Sir.
Okay, everyone with me so far? সবাই ঠিক বুঝতে পারছো? You are here, right? Okay, I hope so. That we are here. We have changed the row names to gene IDs, and deleted everything other than the conditions and the different number of transcripts. This is how we prepare the dataset to put it into the DE-seq2 tool. Okay? So now next, if you want to figure out key, yes, there are 506 transcripts of the first, 192 of the second, 913 of the third, but how many transcripts total did I actually find out from my sequencing? So how many mRNA transcripts are there in total, which is essential addition of all of this, nothing else. So you do this to figure out that your this is known as depth of sequencing, how many transcripts you are able to find out from the sequencing result. And if all of these you obviously get the idea that if all of these rows don't have similar number of transcripts, and one of them has ridiculously less or exceptionally more, then there is something wrong. And in particular one condition, you are getting 510 times more mRNA than anywhere else. So you understand there is something wrong with this particular data set, maybe you should not proceed. So for that, what we do is that we find out the library size, and we do that using the vector mute.data lib size, use the function column sums, which just adds the column, nothing fancy, and then divide by 10 into e to the power 6, so that we get the value in a million. So we then plot it, bar plot mute.data lab size, and then add just name to the y-axis, library size per million. If you do that, you should see a very simple graph which looks like this. So for replicate 1 we have like 3.8 million reads, replicate 2 you have like 4 rectoconjugation million reads, due to wild type are exactly almost the same, around 3.8 million reads. Do this and see if you're getting the same thing.
স্যার উপরে একবার দেখুন।
উপরে কোথায়? This is the code না?
ও আচ্ছা আচ্ছা, হ্যাঁ ঠিক আছে।
Okay, did everyone get the bar graph?
Yes, sir.
Okay.
Sir, I have a question.
Yes.
Sir, in this code, 10E6, what does it mean?
10E6 essentially you are dividing but by 1 million. So essentially the column sums would have come around, so here you see it as around 3.8 the value. So actual value is 3.8 million. So instead of just having a huge high bar graph, you divide by 1 million. So essentially you're saying that, how should I explain this? Say you had 3000 transcripts, you divide by 1000, so here we are divided by a million, and then you say like there are 3 transcripts or 4 transcripts total. So essentially it makes the bar plot easier to make, and you are doing it per million. So essentially this value is 3.8 million because you are dividing by 1 million before starting. So this is how you generally plot this. Is that okay?
Okay, sir. Yes, sir.
Next, so DESeq2. So we have prepared our data file called mutedata.deseq2. Next, we have to tell the dataset key what exactly are you trying to do. Here, what we are trying to do is that we have two different conditions: mutant cells versus wild type cells, and we have two replicates of mutants and two replicates of wild type. But you have to tell it to DESeq2, like exactly this is the idea that you are going for, because for the computer it is just numbers after numbers. So in order to tell it this is the idea that you are following, you make a mute data dot call data frame. So this data frame gives the information to the program that yes, this is my idea. So what is the idea? So idea is, how you do it is that you make a data frame. So data frame just makes a data frame, and you say that first column is called condition, which has factor variables mut, wild type and wild type, and then you have replicate as the second column, which is as factor 1 to 1 2. And if you just display it, it tells you the same thing that I just told you. So you have the conditions are mutant and wild type, and you have two replicates of each: replicate 1, replicate 2, wild type, replicate 1, replicate 2. So it is very simple to do in this easy of a data set, where you only have two replicates for mutants, two replicates for wild types. But if you imagine complicated data sets, say you are looking at genes being expressed between the wild type tissue, which is normal completely, between breast cancer and colon cancer, and under different conditions, then this this mute data dot call data frames becomes very complicated. You are looking at different like 3 different conditions, and in those conditions maybe you are looking at, if I treat with drug A, what happens, if I treat with drug B, what happens. So this becomes very complicated. But here it is pretty simple. You just put in the different conditions that you are looking at and the replicates that you have. So different conditions: mutant, wild type, and replicates are just 2. So replicate 1, replicate 2, replicate 1, replicate 2. Essentially, whatever you had here, this part of it, you are just writing that down as the conditions in the call dataset. So write this down and just display it. You should see a data frame which looks very same to this. সবাই বুঝতে পেরেছো তো যেটা আমি বলছি, যে you just are making sense of the data for the program, and you are just telling it that, see, this is the data you received, and this is what the columns mean. The columns mean that I have mutant 1, mutant replicate 1, mutant replicate 2, and wild type replicate 1, wild type replicate 2. That is all we are doing here, making a data set to make the program understand what we are trying to do. Okay, everyone done with this?
Yes, sir, done.
Okay, so after you have got mute data.call data, all that is left is just running the program. To run the program, you have first we make a matrix out of all the data that we have. So the matrix is called DDS, which is the DE-seek dataset from matrix. So this is the function that you will call. The count data is the actual transcript numbers that the dataset that we made. The column data dataset that we made is just the one before, mute.data call data, and the design of the experiment is
স্যার স্ক্রিনটা একটু উপরে করবেন, আগের কোডটা একবার দেখবো।
হ্যাঁ দেখো,
হ্যাঁ স্যার হয়ে গেছে।
so you count data is your transcript numbers, call data is the data from the columns that we just made, and design is the condition. So condition is mutant wild type, nothing too difficult here, and you just put your that squiggly line that you find above your tab key on your keyboard followed by condition. So this will make you a matrix which is all of this data combined, and once you have made the matrix, you add the data of the gene names. So you remember the gene names that we removed, so we can add them at this point, so that later on it is easier, just you don't look at the ensemble ID but the gene names. So you can add row data DDS equal to mute data $sign gene name, and then just display DDS. Go ahead and do this and see what you get.
স্যার বলছি আগেরটা না আমার একটা এরর দেখাচ্ছে, একবার স্ক্রিন শেয়ার করছি।
হ্যাঁ দেখো, it is
দেখাচ্ছে
data frame arguments imply different number of rows. Why can't I see properly? The frame condition equal to factor replicate equal to as dot factor C1 2 1 comma 2 comma 1 comma 2 dot নয়। So when you comma the spelling is wrong. Your mute data dot call data দুটোতে d গুলো ক্যাপিটালে, হেডে ক্যাপিটালে নেই। Yes.
ওকে স্যার।
When you put in 1.2, it is imagining that you have 3 numbers just. That's why it is giving you error. Okay, coming back. Everyone done with this? Just go and run this. You should see that it has made DDS matrix, which will give you output such as this. The class is essentially the program that we're using, DESeq2. Dimensions, you have 32,000 different essentially gene names, and then 4 columns after that. Row names are the ensemble IDs, row data value. Call names are our names of the column that we initially put in, and data.names is condition and replicate. Everyone got this?
No, sir.
Okay. Like if you're not getting an error, then no.
Sir, if you could wait a bit.
Yes, I am waiting. You just go ahead and try.
Sir, I'm getting an error in the first line of the code. DDS equal to disk.
Check if the spelling is correct. If the spelling is also correct, then just show me your screen, and we can look at it together.
Sir, I want to share the screen,
Yes,
then
yes,
I
yes.
think.
Share your screen. Let me also look at it.
Okay.
Okay. It is evaluating argument X. Selecting a method for function and call. Okay. I will remove the banner in the bottom that you have. Meet.google যেটা আছে hide করে দাও ওটাকে. I want to read what is on the console. Object mute data.deseq2 not found. Okay, go up. I think you have not run one of the blocks. Go up. Go up. Run this. Aha, you see, you have saved mute data.deseq as DESQ, not DESEQ. তার জন্য when you're putting that in the last one, it is not able to find anything.
Okay, sir, understood. Thank you.
Do that and see if you're getting the result.
Yes, sir, got it. Thank you.
I hope everyone is on the same page, that you get output similar to this, in which you see that essentially your DDS matrix is prepared. Once the DDS matrix is prepared, you are home free. The pre-processing of the data is done. You can just run DESeq2. I have already done this, I guess. This is already done. No. What? No. Stop. yes, once this is done, you go to actually, so this is the chunk we are looking at, not this one. So you just run the deseq2 function. So dds equal to deseq, and then bracket, inside the bracket dds. It should just run deseq2 if we have done everything correctly, and the results, to find the results of the entire thing, you write res is equal to results dds, and then display res, and res $sign gene name equal to row data dds $value, so that the gene names gets replaced by the value. Just go ahead and do this, and you should find stuff happening, like the entire process starts. Essentially, this first line is the most important. You call the function and tell them to analyze your matrix that you have prepared, and then just display the results. You should see that the console is doing stuff, and then it gets done. Tell me when everyone has run this.
Done, sir.
Okay.
Sir, for me, it gave a result of up to the line fitting model and testing. Uh, After that, there is nothing.
Yeah, because it is fitting the model and testing, it will give you the did you do this, did you just display this? It either is
Oh,
still processing it, or it has processed it, and you have to display the results.
it displayed the results like after that again.
Okay, so now let us look at what results we got. Yes, so log to fold change. So when we are comparing two different levels of gene expression, say any gene A, it was like we had 100 transcripts in condition A and 500 transcripts in condition B. How we look at it is, we only care if the number of transcripts has either become 2 times the control value, or 2 times less than the control value. Yeah, it either increases 2 times, or it decreases 2 times. Our idea is that then only there is some practical change in gene expression. If it is less than 2 times change, we actually don't care. So how do we calculate that? What we do is that we calculate something called log 2 fold change. So we know what logarithms are. Logarithms, whatever the base is, so x equal to 2 times a, log x equal to a log 2. So when you do a log 2 fold change, you essentially change all the fold change is essentially the division. Or let me just write it down for you, it is easier. So fold change is equal to mutant condition mRNA transcripts divided by wild type condition mRNA transcripts. This is for all the genes. So you just divide both of them, and In mutant conditions I had 500 transcripts, in wild type conditions I had 100 transcripts, so fold changes 5 times. My gene expression increased 5 times. Excellent. But as I said, we only care about if it is more than 2 times, so we do something called log 2 fold of the fold change. When you do log 2 of the fold change, you get essentially, in our example, log 2 of 5, which is greater than 1. If it is greater than 1, then we know that yes, the fold change is at least twice or more, and then we just look at values which are greater than 1. Values which are less than 1 we don't care of. But interestingly, it also will matter if our log 2 fold change is less than 1. Less than 1, what does that mean? Less than 1 will mean that the log 2 fold change is at least 2 times less or lesser. So as I said, we care if the mRNA number of transcripts is either has increased 2 times or more, or has decreased 2 times or less. So you know that how log works. Whenever you have a decimal point thing, then it gives you minus values. So anything, the values of log 2 fold change, if it is less than -1, then the gene expression level has decreased 2 times or less. If it is greater than 1, then it has increased 2 times or less. So we only look at values which are either 1 or more than 1 or less than -1. Is that okay?
Sir, can you explain once more?
Okay. So as I said, we, in case of analyzing the data sets, we care if a particular mRNA's number of transcripts has either doubled or more. So say originally there were 100 mRNAs of that particular gene, and I will care if either it has become 200 or more, or it has become 50 or less. So either it has doubled in number and it is more than doubled in number, or it is less than halved. So it is either less than 2 times the total control or wild type value, or it has doubled from the wild type value. So essentially 100 transcripts originally, I care if it has become 200 or more, or it has become 50 or less in my condition B or in the mutant condition. That is very easy to find out. How we do is that we just find out what is called the log 2 of the fold change. So fold change is essentially the mRNAs in the mutant conditions divided by the mRNAs in the wild type conditions. So fold change would be just the ratio of the number of transcripts in mutant conditions divided by the transcripts in wild type condition. When you do log 2 of the fold change, you actually find out if the value has either doubled or more, or it has become less than 2 times the value. We actually don't care about anything about the mathematics. You just run the log 2 fold change function, which DE-seq2 automatically does for you, and you look for genes which have either a value of 1 or more, or a value of -1 or less. If it is 1 or more, then you know that there has been a lot of increase in the mRNA, and if it is -1 or less, there has been a lot of decrease in the mRNA. Both of them are interesting to us because then there is differential gene expression. So essentially, if you look at the table, there is a log 2 fold change table, and you look for genes which are 1 or more, or -1 or less. So this is an interesting gene. This is a very interesting gene because it has reduced a lot, and you look for these values. These ones you don't care of, the ones which are between 1 or -1. Everyone okay with this?
Yes, sir.
Okay. Now there is a p value for all of this as well. So it has found out what the fold change is, but it also has found out if it is significant or not. This is the same value, same thing that we all know, that if the p value is less than the p-value is less than 0.05, then we care about it. If it is not, we don't care about it. So values such as this is important, values such as this is important, but this not so much because this is not significant. So you take a look at both of these things. You look at log to fold change if it is greater than 1 or less than -1, and the p-value. The log to fold change tells you that there has been a practical differential gene expression here, and the p-value tells you if this fold change is actually trustworthy or not. So if you see a 10 times fold change and you're like, this gene is the best in the world, but then the p-value is something like this, which is not significant, which means that the calculation for the fold change is not correct. So you don't, even if it is a 10 times change, the p-value is not significant, you don't care. So you look at only the ones in which the p-value is less than 0.05 and the log to fold change is greater than 1 or less than -1. Is that okay with everyone? বুঝতে না পারলে
Okay,
প্লিজ
sir.
বুঝতে না পারলে কেউ প্লিজ বলো. So essentially I'm saying conditions for a differential expression of genes is the log to fold change -1 থেকে কম or 1 থেকে বেশি. Actually I am putting the arrows wrong. Am I putting the arrows wrong? So it should be, how should I put it?
So একটা কোয়েশ্চেন আছে, আই থিঙ্ক আমি একটা বেসিক আইডিয়ার আন্ডারস্ট্যান্ডিং ভুল হচ্ছে আমার।
হ্যাঁ হ্যাঁ বলো.
আমার কোয়েশ্চেনটা এটা ছিল যে আমরা বেসিক্যালি এখানে মেজার করছি চেঞ্জটা, এটা হচ্ছে যে মানে ওয়াইল্ড টাইপে কতগুলো এমআরএন প্রডিউস হচ্ছে ভার্সেস মিউটেশনে কতগুলো এমআরএন প্রডিউস হচ্ছে, তাই তো?
Yes. Wild type how
তো
many?
যদি তাই,
হ্যাঁ বলো.
তো যদি তাই হয় যে শুধু নাম্বারটাই ক্যালকুলেট করছি, মানে নাম্বারটা তো ওই ডেটা সেটে র ডেটা সেটে পেয়ে গেলাম যে
Exactly.
ওয়াইল্ড টাইপ এত রেকর্ডেড আর মিউটেশন এত রেকর্ডেড, তো হচ্ছে যে ওটা রেকর্ড করছে যে ডিফারেন্সটা এসেনশিয়ালি কত, মানে কত ম্যাগনিটিউডে মাল্টিপ্লাই হচ্ছে ইয়েতে মিউটেন্টে, সেটা বুঝলাম ম্যাম। বাট সরি স্যার, বলছি বাট পি ভ্যালুটা তাই পি ভ্যালু ভুল কি করে আসতে পারে, আমি ঠিক বুঝতে পারি ইনঅ্যাকিউরেট কিভাবে আসতে পারে? তো সিম্পলি ক্যালকুলেশনের ব্যাপার না, যে বেড়েছে কি কতটা কমেছে লগ টু ফোল্ড চেঞ্জ দিয়ে, লাইক পি ভ্যালুটা ইনঅ্যাকিউরেট অ্যাকিউরেট কি করে আসবে?
হ্যাঁ, সো এখানে ফোল্ড চেঞ্জটা ইভেন দো দ্য ভ্যালু ইজ জাস্ট দ্য ডিভিশন অফ দ্য টু, যে টেস্টটা করা হচ্ছে, যেমন আমাদের টি টেস্টের সময় তো উই স পি ভ্যালু ইভেন দো উই আর জাস্ট লুকিং এট দ্য ডিফারেন্স বিটুইন দ্য টু. So এখানে when we are actually doing the DE-seq2 test, it is just not dividing the two. Even though this fold change is this division of the two, it is doing a number of other statistical analyses to figure out if this fold change is trustworthy or not. There is a lot of things going on in the background. Even though this value is something that we can calculate right off the bat, we just divide the two of them, we know it. But there is a lot of other things that happens. There is normalization that occurs that DE-seq2 does by itself. It fits it on a regression curve to figure out that this thing is trustworthy or not. It estimates like builds a network between the genes, so it does a lot of things. So p-value essentially tells you much more than what you will get from just dividing. That is why we are not just doing like extracting the two values and just dividing them and finding out the fold change. DE-seq2 actually use quite important. It is doing a lot of things.
Okay, sir. Then DE-seq2 is it checking the validity of the data set, or is it checking the accuracy of the results that we have got?
It is testing the accuracy of the results, but it is also fitting a regression model on top of it. So it is essentially যতটা আমরা এখন অবধি করেছি, টি টেস্ট করেছি, linear regression, multiple regression, logistic regression, DE-seq2 does all of this and does much more. Just it has been dialed down so that people who are not very much interested in the mathematical part of the statistics can just look at two things and figure everything out. So we only care, it is very easy for us to just look at this, but I am not also going into that part because it is very complicated. But we just care about this. DSeq2 does it very easily for us. ठीक है sir?
Okay, sir. Thank you. Thank you, sir. Got it.
So yes, conditions for differential gene expression is log to fold change greater than 1 or log to fold change less than -1, and the second one is p value less than 0.05. That is as easy as everything that we have been doing so far. So these are our conditions based on which we will say that our gene is differentially expressed or not. So you see that this data set looks a bit complicated, and there are some values called NA. NA means non-numeric values. So these are in cases where either in one of the conditions there was no data, or the data was a rubbish data, so DSeq2 just doesn't put it into the data set. We don't want it in our final results, so we do something called cleaning up the entire thing. So how do we clean up? We first make this entire matrix into a data frame, and then run our age old helpful select and filters. So first we do is that we only care about, as I said, the log to fold change, the adjusted p value, p adjusted, and the gene names. We don't care about the base mean, the LFCAC, the stat, the p values, original p values. We don't care about those. We care only about the p adjusted, log to fold change, and then what is the name of the gene. So we only select these, and then filter for p values less than 0.05, which we actually care about, and then filter for log to fold change, which is greater than 1 or less than -1, also what we care about. And now we store it in the dataset result.deg. So this final dataset will contain everything we want. It contains the gene names which are differentially expressed, how much that differential are they using the log to fold change data, and if they are significant or not using the p adjusted data. And finally we should and you do a head of entire thing, and you finally find that this is the dataset you get. You find the ensemble IDs, how much the change is, the p adjusted values, and the gene names. So essentially using this we have finally done our RNA sequencing data analysis. We have found the differentially expressed genes which are differentially expressed as well as significant. So just run this. হয়েছে? Everyone got the results?
Got it, sir.
Okay, everyone else?
Sir, can you explain the last line, that is dim result
Yes,
dot
so
DG?
when you do dim for any data frame, it gives you the dimensions of the data set. So imagine ki how many rows and how many columns. So if you just look at it, so there are 938 rows this value is important because it now tells you the number of differentially expressed genes. So the rows, every one of the rows has the gene names, right? So if you just count the number of rows, so you can find the differentially expressed genes. So there are 938 differentially expressed genes in the entire experiment that you have done. So it helps you determine that what the situation is. So this is how you do it. And finally, I will just give you this in the chat. So run this. Yeah. It seems no. Okay. Yeah. Don't run the whatever is under hashtag prepared data. We have already done that. You just delete that line and run everything else.
Sir, can you show the previous code once again?
Previous code, yes.
Yes, thank you, sir.
ঠিক আছে,
স্যার কোনটা রান করবো না?
এই hashtag prepared data you don't have to run. We have already done this. We just you just run everything else other than that. Copy paste and hashtag prepared data under everything delete, and then run it.
স্যার আরেকবার বলবেন কোনটা রান করবো, লাইক
Whatever you see under the so you just copy and paste whatever I have given in the chat,
yes
and hashtag
yes
prepared data পরে যে দুটো লাইন আছে about res_df, these two lines you
yeah
delete and then run everything
okay
else.
okay
Just এই দুটো লাইন you have to delete, don't have to do anything else, just run. Done, everyone?
sir, can I share the screen? It's not coming, so can I share it?
Yes, you can share your screen. Just show me what is the problem.
Sir, can I see?
Not yet. Yes, I can. Error, comma in API.
Sir, here I guess.
Delete. I told you to delete everything. Prepare data niche সব কিছু delete. And then
Okay.
run. Result.dg not found. Did you not run the previous chunk? Run the previous
Okay.
chunk.
Okay.
You have to run the previous chunk, na? Nahle to it won't initialize what the result of that thing is. What is this? Result.deg. So that we found result.deg in this section. You have to run this and then it will work. Okay. So essentially, yes. So we have done all of the things. We have found the result, but then we want to visualize what the results look like. In order, in order to visualize the results, we plot it as a volcano plot. So it looks like a erupting volcano. So it would have been better if there were more things here. Most of the times there are in this dataset, there aren't, so it's okay. So in the volcano plot, you plot log2 fold change on the x-axis and minus log10 p-value on the y-axis. And you are very interested in things that are further away from the center towards a bit up. So anything in the y-axis above actually has a significant p-value because we are plotting minus log10 p-value. So minus log 10 করার পরে it's essentially whatever however positive above the x-axis the data is, it is more significant, and however further away from 0 the data is, it is more fold change. So you care about things that are up and away, so around this area. So this gene which will be here, this gene, this gene, this gene, this gene, this one, this one, this one, these are the ones that are you will be most interested in. So volcano plots are a very beautiful representation that you use always to figure out which genes you should follow up on, and just visualize the data at once and see, yeah, these are the ones that I'm interested in. The ones over here, you will see a more very like a lot of genes here. These you are not very interested in. The fold change is very less, significance value is also very less. So this was what I was saying, that most of the genes will be the same between the test and the control, mutant and the wild type data sets. Only some of them will be interesting, and these are the ones which are like above here. If you look at more and more data sets, you will see volcano plots most of the time look like these huge thingies with all the stuff here. I can show it to you. Yeah, if I just do it for res instead of resolution. Yes, let's see. Does it run? Yeah, it still doesn't look very nice. Oh, it's fine. Yeah, so this is what the volcano plot looks like. This is the visual representation of the analysis that we did, and we go after genes that are all over this area, which is like a bit above the x-axis, on the like up and away from the 0. Any questions anyone has?
Yes, sir.
Question আছে বলো?
স্যার কয়টা কোয়েশ্চেন আছে আমার? একটাতে স্যার জিওমেট্রিক পয়েন্ট আলফাটা কি? আলফা ভ্যালু
কোথায় আলফা ভ্যালু?
এই আলফা 0.6 আর সাইজ 1 পয়েন্ট, সাইজ না বুঝলাম পয়েন্টার সাইজ, বাট
Oh, these ones. This is only for how good the value plot looks like. You can change it according to you, and then it will just change something. It doesn't matter what it is. It is just to make the plot look very nice.
আচ্ছা ওকে স্যার। স্যার আরেকটা জিনিস হচ্ছে যে এখানে গ্রে আর রেড লিখেন, তো রেডটা কিসের জন্য?
Red is for like much better values should have red, but we don't see much red. If you look at it, that some of them are the color of the gray is a bit different in between things. So if you had very significant and very good p values all over here, here, then those should have appeared as red. But since we don't have any data like that, so we don't see any reds.
আচ্ছা আচ্ছা, লাস্ট কোয়েশ্চেন। বুঝছিলাম যে পি ভ্যালু এখানে মানে মাইনাস লগ 10 অফ পি ভ্যালু যেটা পেয়েছি আমরা, এটা যত বাড়ছে তার মানে পি ভ্যালু মানে তার মানে এসেনশিয়ালি মানে তত বেশি অ্যাকুরেট, রাইট?
Yes, যত p, the original p value is say 2 into 10 to the power minus 2. So when you do a minus log 10 of that value becomes positive and large. So the lower the original p value is, the more positive and larger the minus log 10 p value is.
আচ্ছা আচ্ছা, ঠিক আছে, ওকে স্যার।
Okay, so yes, this is all I had for you today, and I think in the entire course. But I would like to congratulate you. You have actually by your hand done the analysis for a RNA sequencing dataset. Like this will help out a lot in actually figuring out how to do stuff, and I am pretty excited about how you will use all of these skills later on. I just want to tell you that I have not gone through another type of sequencing that I think you should look at once, which is called single cell RNA sequencing. The idea is the same as bulk RNA sequencing data that we have looked at, but you look at individual the mRNA from individual cells. You can actually find out this cell was the one which was describing this mRNA, this cell was the one which was describing this mRNA. So I'm not covering it in the course, but there are multiple YouTube lectures on these. you should just go ahead and look at it once, because this is the technique that everyone is using nowadays to figure and answer a lot of questions in biology. So just giving you a heads up, if you're interested, you can go and look at it. So I will share all of this with you. So in this entire course, we have actually gone through the basics of R, the how to make a vector, how to subset, how to use different small operations, followed by introduction to statistical analysis, and how to use basic statistical analysis such as t-test, normal distribution, ANOVA, to figure out differences between data sets. And then we talked a bit about inferential statistics, how to use probabilities to find stuff out, and finally we ended with actually a practical session on how exactly to use all of this knowledge that we gained in order to solve biological problems, looking at how you analyze a bulk RNA sequencing data, which used all the knowledge that we have gained from throughout the entire course. This course would not have been possible without the help of Professor Arjun Ramkrishnan from the Department of Biological Sciences and Bioengineering, IIT Kanpur, who was kind enough to share his notes with me so that I can use them to teach everyone. And also the book Learning Statistics with R by Daniel Navarro, absolutely excellent. You guys should go ahead and check it out. Completely free resource, and you can learn more about statistics from it. Okay, then I will just quickly take your attendance. Does anyone have anything to say finally?
Thank you, sir, for taking the class. I have never thought that coding, quote unquote coding, can be such interesting uh, in terms of art. I have just thought that it will be the same Gisapita coding and stuff that will be boring, and I'll be here only for attendance and certificate. But it was pretty much interesting.
That's very nice. Yes, thank you. You guys have also been very
Sir,
diligent.
thank you, sir, for taking our classes. Sir, these courses will be available in the YouTube, right?
Yeah, all the data, all the videos that have been recorded are available on YouTube. I've been uploading it side by side with the classes. I think with this, I think all the if I upload it today only, you will find the entire course on the my YouTube channel. Yes, and you have access to the R notebooks, I believe, from the Google Books. You have everything that you will need.
Okay, sir. Thank you, sir.
Okay, so final thing. So we still have the assignment that you guys have to submit today, and I have to think of what to do about the final test, which I will talk to Professor Riddhi about, and then I will inform you. You will get a mail about the final test, which would probably, if it is a final test, then it will probably, I will schedule it for next Thursday, Sunday, but you will get ample warning of this. And if it is an assignment, I will just give the assignment to you. We will have a week to solve it, same as the one that we are doing now. Probably the groups can remain the same. If you guys are okay with it, I will talk with Professor Riddhi and let you know. Okay, thank you everyone for joining. Best of luck.
Thank you, sir.
Thank you, sir.
Thank you so much, sir.